Hello, my name is Brad Frecking. I'm a research geneticist with the USDA ARS located at the U.S. Meat Animal Research Center in Clay Center, Nebraska. And it is my pleasure to represent the Sheep Gems team for this webinar on the topic of genetic conditions. If you have not already done so, I would hope that you will first take the time to view the introductory presentation by Ron Lewis that gives an excellent overview of the project objectives, the scientific team involved, and the contributions from the industry and funding agencies supporting this effort. Much thanks to Ron, so we do not have to cover that information again. I do want to recognize how Sheep Gems is being supported, and with tremendous industry interactions, the group was able to win a grant funded by the USDA National Institute of Food and Agriculture to support this specific R&D project. As pointed out in Ron Lewis's presentation, a key aspect of the Sheep Gems project is to incorporate genomic technologies directly into our genetic evaluation system through NSIP. The current commonly used genotyping chip assay has nearly 50,000 individual single nucleotide polymorphisms, or SNPs, in total. This assay chip was the combined outcome of international collaborations that selected SNP markers for their genetic location, their intermediate frequency, and the ease of genotyping. Additional markers have been added to the assay chip to achieve parentage exclusion objectives and to report specific genetic conditions. Now, just like we want our lamb consumers to know where their food comes from, I think breeders and producers should know to the extent possible where their genotypes come from and that they are not all the same in terms of quality. Now, this does not require going too deep into the chemistry. But this first panel represents how samples would look like for a summary of results from roughly 650 individual sheep genotyped for a single SNP operating normally with two alleles. One allele is represented as a red colored dot, and the alternative allele at that SNP is represented as a blue colored dot. And when an animal is heterozygous, containing both alleles, it is represented in the middle purple distribution. Now, the second panel represents an SNP marker that is not functioning quite as well, and as you can see, the distribution of signals from the red dots is much wider and makes it much more difficult for a computerized system to classify what makes a specific genotype call. So, unlike the SNPs for genomic predictions, where we can just eliminate or screen out those poor performing SNP in our analyses, for genetic conditions, each and individual very specific SNPs are a must-have. This unfortunately leads us to have some unreported calls for some animal samples. Now, genomic tools in the form of these SNP genotypes allow us to achieve three important aims. The first being validation of the pedigree that's recorded. And the second is to allow more reliable predictions of genetic merit for complex traits like robustness and climatic resilience that Sheep Gems is attempting to do. And finally, the topic for today's discussion, to generate genetic condition status for some important fitness traits such as ovine progressive pneumonia and scrapie susceptibility. As part of the Sheep Gems project and other prior historical genotyping efforts associated with NSIP has allowed us to summarize genotypic data on more than 16,000 individual U.S. sheep. Now, we realize that there are additional genetic conditions that producers may be interested in, and there are certainly more than these 5 represented on this 50K chip. But these are the 5 that have currently gone through a complete validation process as we evaluate genotypes coming in through Sheep Gems and NSIP. Now, this is a very different approach than directly requesting these specific genetic tests from a commercial provider. Now, we understand that some producers may want and or need this genetic information at different time points than what is currently provided by this pipeline. However, each individual producer must evaluate the needs versus cost of the information. The information provided in sheep gems is essentially value-added information from what is needed for genomic breeding value estimation. That value-added information would cost an additional $15 to $20 per animal sample for each additional genetic test, as well as parentage. So for the $25 per sample cost being charged, you are conservatively obtaining roughly $100 to $120 worth of additional value in genetic testing above and beyond the genome-enhanced breeding values. Now, the conditions currently being reported are scrapie susceptibility, susceptibility to ovine progressive pneumonia, myostatin double muscling, Calipige muscle hypertrophy, and the fifth one is the variant responsible for Borrelia fecundity. Now, fortunately, all 5 of these conditions are on different sheep chromosomes, which means that all the information can be used independently from each other. These 5 conditions have been validated on the 50K genotyping assay by blindly submitting to the commercial lab a set of 15 previously known genotype samples that cover the diversity of the genotypes that we would expect as standards for comparison, and all have been returned to us with 100% concordance. The first genetic condition, scrapie, is a degenerative disease affecting the central nervous system and can be debilitating to a flock. Once an animal is infected, there is no treatment, and infected animals typically don't show symptoms until 2 to 5 years after their initial infection. After the onset of symptoms, animals tend to live only 1 to 6 more months. And infected animals, when they begin to show symptoms, include nervousness or aggression, obsessive rubbing, incoordination, and tremors. Because of the delay between infection and onset of the disease, control of this disease within a flock by signs and symptoms alone is ineffective. The ability to manage a flock using genetics has made it increasingly easy to prevent scrapie. 2 DNA locations that code for the prion protein are particularly important for classical scrapie susceptibility: codons 136 and 171. Codon 136 codes for the amino acids alanine A or valine V. Codon 171 codes for the amino acids arginine R, glutamine Q, histidine H, or lysine K. Now, for selection purposes, the only known resistance is for the R amino acid at codon 171. Animals with the combination of AA at codon 136 and RR at codon 171 are completely resistant to the disease. This means that while other animals in the flock may have the disease, these animals will never become infected. However, any animal with the QQ genotype at codon 171 will be susceptible to classical scrapie, while other codon combinations grant varying levels of partial resistance. The genetic condition status of the prion protein gene differs appreciably among Katahdin, Polypay, Rambouillet, and Suffolk breeds. In our data set, most, or approximately 92% of Polypays were deemed completely scrapie resistant, represented by the dark green bars. The majority of Katahdin at about 93%, Suffolk at about 92%, and Rambouillet at 84% are either resistant or rarely susceptible, which is a combination of the dark and light green bars on this graph. However, 15.8% of Rambouillet were characterized as highly susceptible to scrapie, which is represented in the red bars. Selection to increase the amount of scrapie-resistant genotypes across breeds and the nation is considered a key part of the national scrapie eradication effort in the U.S. Ovine progressive pneumonia, or OPP, also known as Médy Vizna outside of the U.S., is an infectious disease of sheep caused by the OPP virus, a very slow-growing small ruminant lentivirus. The disease itself, as opposed to positive infection status measured in the blood serum, as it progresses, the disease status is eventually fatal. General wasting or thin ewe syndrome, decreased milk production, usually hard bag syndrome, and pneumonia that is unresponsive to treatment with labored breathing, exercise intolerance, and coughing are the most prevalent visible signs. Symptoms are not apparent until later in life, so the infection status can only be determined by testing. Depending on the incidence of OPP in your flock, production losses can be significant. There is no known treatment and no vaccines are available. Variable degrees of natural resistance to infection, however, do exist and can be determined by genetic testing using variation at a gene called TMEM154. Selecting replacement breeding animals carrying desirable genotypes can help decrease susceptibility to OPP in the flock. Now for TMEM154, the 4 most common genetic haplotypes, one inherited from the sire and one inherited from the dam at TMEM154 are denoted 1, 2, 3, and 4. And those 4 have been experimentally validated in animal populations. The haplotypes shown to confer less susceptibility are designated 1 and 4, as shown by the green values in the stoplight of the figure. Therefore, diplotype combinations, or 1-1, 4-4, or 1-4 genotypes, are the most desirable when selecting animals for breeding. While haplotypes 2 and 3, even in combination with some of the other more desirable haplotypes, indicate high susceptibility and are less desirable, as represented by the red values in the stoplight. We recommend that producers increase the frequency of haplotype 1 as part of their existing breeding program. If producers have access to replacement animals with haplotype 4, it can be selected at the expense of haplotypes 2 and 3, but not at the expense of haplotype 1. The rationale for the last recommendation is that haplotype 1 was just as effective as haplotype 4, yet it is only one amino acid different from the common ancestral protein sequence, which is designated haplotype 3. Thus, haplotype 1 has the best chance at retaining what we would consider the normal natural function of the TMEM154 gene or protein, which is currently not well understood. No diplotype at TMEM154 confers complete resistance. So if OPP-free flock status is important to you, it is recommended a sample of your older ewes be tested periodically using a blood test, an OPP ELISA test that detects the presence of antibodies against the virus. Control of OPP consists of the selection of naturally less susceptible animals for breeding and a test and cull scheme where seropositive animals are considered infected and removed from the flock. Genotypic status at TMEM154 differs substantially among Katahdin, Polypay, Rambouillet, and Suffolk breeds in the U.S. In this figure, the less susceptible combinations of alleles are represented by the dark green bars, and the susceptible combinations are represented by the red bars. Nearly 60% of Katahdin and Suffolk sheep contained at least one of the highly susceptible alleles relative to OPP. On the other hand, in Polypay, only 8%, and in Rambouillet, only 15% were highly susceptible. It may be tempting to use the TMEM154 genotype as a primary selection criterion. However, we'd recommend a more balanced approach where this information is incorporated as part of an existing breeding program. This allows breeders to continue to make genetic progress on their current priorities while still reducing OPP genetic susceptibility in their flock. One reasonable approach, with all other things being equal in your selection criterion, is favoring sires with the more favorable haplotypes of 1 and or 4. Myostatin, also known as double muscling, is known to have originated and occurs frequently in the Texel or Beltex breeds of sheep. Normally, ancestral myostatin alleles function as a negative regulator of muscle growth. And the impact of this specific genetic variant in Texels results in reduced myostatin protein levels, which leads to then increased muscle growth, primarily reflected in increased fiber numbers, in addition to some fiber hypertrophy. Animals having one copy of the variant have been known to exhibit intermediate, or partial double muscle phenotypes. Lines of animals within breeds that are targeted for use as terminal sires would be the most logical component in our production systems for increased selection for these myostatin alleles. The frequency of the myostatin mutation, which originated in the Texel breed, was small in these four populations, as one would expect. In Katahdins, 4.3% and 0.4% of the animals carried one, represented by the red bars, or two copies, represented by the dark green bars, respectively. In Suffolks, those equivalent values were 6.2% and 0.2%. This myostatin mutation was not detected in Polypay or Rambouillet breed samples. The causative mutation for this genetic condition occurred in a single Dorset ram and has dramatic effects on muscle development. The genetic locus was given the descriptive name callipege, which was derived from Greek for beautiful buttocks. The phenotype is characterized by extreme muscle development due to hypertrophy during early postnatal growth, 3 to 8 weeks of age. Despite the large favorable effects of this hypertrophy condition on slaughter and carcass composition traits, the associated adverse effects on meat quality traits have deemed this a mutation to eliminate for the overall good of the U.S. sheep industry. Due to the unusual inheritance of the callipege phenotype, it is important to realize that the genetic testing will identify the number of copies of the mutation, but will not predict the phenotype without additional parental status information. Regardless, that test can still be used to remove the callipege mutation from the existing populations. The last genetic condition we will cover, the Borula fecundity gene, or FecB, is inherited as a single autosomal locus influencing ovulation rate and litter size. The effect of FecB is additive for ovulation rate, increasing by about 1.6 corpora lutea, or eggs, per cycle for each copy of the mutation. Multiple researchers identified the causative mutation within the bone morphogenic protein 1B receptor. These teams found that Borula sheep had a mutation at codon 249 in a highly conserved region of this gene. Now, selection for multiple copies of this fecundity mutation does not cause infertility like some other example mutations that affect fecundity, but it does potentially result in large changes to ovulation rate and litter size between the genotype classes, and the existing production system clearly needs to take these potential substantial changes into account. There were no animals in our data set that contained the specific FecB mutation. However, it is interesting that additional variation does exist in U.S. sheep populations, but those variants are not yet on the chip, and perhaps more importantly, have not been evaluated experimentally. Those variations that impact the BMPR1B protein are in codon 64 in Katahdin sheep and codon 345 in Romanoff and Finn sheep breeds. Our thanks is given once again to our supporting organizations to make this effort possible, and more specifically, we received support from multiple national sheep organizations as well as along with breed associations, and we give you our sincere thanks. Your willingness to back Sheep Gems has made a tremendous difference. For any additional questions for this topic, please feel free to contact either myself or Ron Lewis at the emails provided here. And with that, I will now conclude this Sheep Gems webinar on genetic conditions. Thank you.